All right, now my mic should be working. Am I back? Is it back? Is it working? Let me just check it one more time. Okay, yes, we are here. Okay, no one saw that first one. But hi, I'm Millennium Isekai'd Mask Knight Empress Scientist, and today I will be doing a science session because, as you know, at the end of my tags, I am a science lecturer as well. So this might be 30 minutes, this might be 3 hours, who knows. But forgive me if I miss chat because I'll be focusing. Oh, and before I start, look at this rig upgrade. Look at that! The jawline! And it will puff the cheeks. And I learned how to separate the blinking. Wink one eye. Okay. But yeah, let's go. So where should I start? I will start with membrane lipids. So what are they? What are their known functions? How do we study them? So yeah, what's the structure of the membrane? The plasma membrane encloses the cell and it defines its boundaries and maintains the essential differences between the cytosol and the extracellular environment. Okay, so the plasma membrane encloses the cell, defines its boundaries, and maintains the essential differences between the cytosol and the extracellular membrane or environment. And can we see the membranes? No, we can't see the membranes. Why? Well, not in the light microscope, which has a below limit resolution of 200 nanometers, it seems, but it can be seen using an EM resolution around 6 to 8 nanometers. And the cell membrane model, aka the fluid mosaic model by Singer and Nicholson in 1972, it depicts the membrane as a fluid phospholipid bilayer with embedded proteins that can move laterally within the membrane, giving it a mosaic-like appearance. Again, what is this one? The cell membrane model by Singer and Nicholson will allow it. It depicts the membrane as a fluid phospholipid bilayer with embedded proteins that can move laterally within the membrane, giving it a mosaic-like appearance. So, the proteins will move laterally within the membrane, giving it a mosaic. I don't know. And what are lipids? Why are there lipids in rafts? So the lipid in rafts or lipid rafts, the function is to concentrate the receptors for interaction with ligands and effectors. So they are in the rafts because, let me just check something. Yeah, there you go. So they are in the rafts because it's to concentrate receptors for interaction with ligands and receptors. And the plasma membrane contains patches of sphingolipid enriched domains. So the plasma membrane, what does it have? Patches of sphingolipid enriched domains. Okay. The plasma membrane contains patches of sphingolipid enriched domains separated from the remaining plasma membrane by specific phase behaviors. And there are small dynamic microdomains within the cell membrane that are enriched in certain lipids and proteins. So the lipid rafts is essentially just an area in the plasma membrane enriched with different lipids and proteins. How are they maintained? They are maintained by hydrogen bonds, such as sphingolipids. They're maintained by hydrogen bonds with like sphingolipids and cholesterol. So how are lipid rafts maintained? It's maintained by hydrogen bonds and lipid rafts, and cholesterol. And they also they organize numerous receptors or signaling pathways. So what is happening? They are organized by specific receptors. No. They organize numerous receptors by signaling pathways. And it is a local area of membrane where you have phospholipids, sphingolipid, cholesterol, and membrane proteins are concentrated. So what are lipid rafts? It's an area where proteins and lipids are gathered, and it is maintained by hydrogen bonds, and it is a small microdomain within the cell membrane enriched in lipids and layers. Oh, I think I forgot to add what's that called? The thumbnail. Let me add that. Now it's there. Yeah. Okay. Hopefully that works. Okay, next. What are caveolae and rafts? I know, that's a big question also that I want to ask. So what are caveolae and rafts? Well, they are specialized they're also specialized domain in the membrane involved in cell signaling and endocytosis. Okay, so caveolae contains caveolins. What are caveolae or caveolins? They are integral membrane proteins that help stabilize their shape and regulate signaling molecules. And these structures play crucial roles in processes such as signal transduction. So, in endothelial cells, especially in capillaries, caveolae facilitate molecular exchange by transporting lipids, proteins, and solutes across the membrane. They are like mini bowls. So caveolae has caveolins, 1, 2, 3, and they help maintain the shape and regulate the signaling molecules, and they play crucial roles in processes such as signal transduction, cholesterol, homeostasis, and endocytosis. However, in hepatocytes, stuff in the liver, and adipocytes in the fat cells, while caveolin 1 is present, it is unclear if they form the flask-shaped caveolae. Instead, caveolins in these cells could play roles in lipid metabolism and insulin signaling without the distinct invagination. So they don't form the bowl-like thing. I see. So in hepatocytes, liver cells, and adipocytes, even if caveolin 1 is present, they might not form the flask-shaped caveolae and would just play a role in lipid metabolism. And that uncertainty also arises from variability in experimental findings and possible different alternative mechanisms. So I have an image, but I can't show it. So I'll just describe it. Top left would be a scanning electron micrograph showing a capillary network, highlighting the interconnected structure of endothelial cells. Top right would have the transmission electron micrograph showing an endothelial cell's ultrastructure. Basically just you're digging in deeper for the caveolae, caveolins, and whatnot. So again, lipid rafts. So if you compare lipid rafts versus caveolae versus caveolin, lipid rafts are the small microdomains specialized in cholesterol and sphingolipid rich areas, and they exist in all cell types. It serves as a platform for cell signaling, protein sorting, and membrane trafficking, but do not necessarily form bowls if they don't want to. Caveolins and caveolae, they are related. Caveolae have caveolins 1, 2, and 3 maybe, and they are required for caveolin formation in endothelial cells, adipocytes, fibroblasts. Well, caveolin 3, remember, caveolin 3 is only specific to muscle cells. I have to grab a pen. Where is my pencil? I'm back. So, caveolin 3 is only for muscle cells. Oh my gosh. Let's highlight that one. There you go. Caveolin 3 is only for muscle cells. Alright. Good. Next. So, how would I go about and isolate and analyze lipid rafts? That's an interesting question because it is a crucial step in studying membrane microdomains involved in signaling and protein organization. So what I will do, I could do cold detergent extraction for the lipid raft isolation, because we can have test tubes labeled with like wild type no triton and wild type with triton, and with a minus and a plus being like indicating the use of a cold detergent to selectively solubilize the non-raft membranes. Okay, so what am I talking about? You have two test tubes, the left and the right. Okay, one has triton, one does not have triton. So the one without triton, why will this work? Because lipid rafts would remain insoluble and can be separated via the centrifugation after you apply triton. So, and why will they remain insoluble? It's because they have cholesterol and sphingolipids. We said that before. Lipid rafts have cholesterol and sphingolipids. And also, the low temperature prevents disruption of raft integrity, so that's why it should be cold detergent extraction. And the thing is, so once you centrifuge it, you have the supernatant and the precipitate, right? One second, I just want to make sure. After the initial centrifugation step, I want to make sure that I say this right. The heavier solubilized membranes remain in the lower fraction. Lipid rafts migrate to the top fractions because they are rich in cholesterol and sphingolipids. Yeah. Okay, my picture is just not very clear, but okay. You solubilize most of the non-raft membranes, but the lipid rafts may remain detergent resistant because they're insoluble. So the centrifugation after after centrifuging, after you centrifuge the thing, this will remove the cell debris and nuclei. And then the supernatant contains both the solubilized membranes and the insoluble lipid rafts. Okay. If I want to draw that, supernatant would be the liquid thing, right? This one. Supernatant would have the non-rafts plus insoluble lipid rafts. Okay. So how can I collect the fractions? They're usually in the fractions 3 to 5, the lighter ones. but the very, very solubilized membranes will be in the lower fractions. So after that, what do I do? What do I do after the cold detergent extraction? I will do a Western blot. Why? Because I want to check for caveolin 1, flotilin, or the GM1 ganglioside. What is a GM1 ganglioside? I am not sure, and that's where Google comes in. I know that I have to Western blot it. GM1 ganglioside. Okay. It's a type of glycosphingolipid, a key component of cell membranes. Okay, yeah, that's why we want it. Because if we see them, they will give or imply that we were successful in isolating our lipid rafts. Awesome. So the Western blot for lipid raft markers will confirm that lipid rafts are in the correct fractions by detecting our dear caveolin 1, flotilin, or GM1 ganglioside. Good. Okay. For Western blot analysis, I will... Yeah, how do we start it? I will extract and quantify the protein from the lipid raft fractions using a Bradford assay. I could do that. But what is a Bradford assay? And Bradford assay. Where is it? Colorimetric. Quick and sensitive method for measuring protein concentration in a solution. Okay. Quick method to measure protein concentration in a solution. Protein concentration in a solution. And we need that because we're trying to determine how much proteins we have there. Good. So you load this part. So you load 20 to 50 micrograms of protein onto a 12 to 15% SDS page gel. Take note. What is an SDS page gel? I think that is a very specific type of gel electrophoresis. Yeah, because when you do that one, was it like only the charge? It uses an electric field to separate charged molecules, and okay, this is the special one. The SDS, or sodium dodecyl sulfate, would coat wait, hold on, let's go. So the SDS is a detergent that would denature the proteins by coating them with a negative charge. That way, when you do the gel electrophoresis, it's only by size. You only separate them by size. That's good. So again, SDS page versus normal DE. The SDS detergent is a detergent that will denature the protein by coating them with a negative charge. That way, the bands are just traveling through the gel because of their size difference, with the smaller fractions traveling faster throughout the gel. There's also another thing. Oh yeah, just a second. Okay, but if we don't do the SDS, then it will travel because of its charge also. And how does it do that? The DNA would have like a slightly negative charge because of its phosphate in the backbone, and so it will travel from the negative electrode towards the positive electrode. Beautiful. Okay, so after electrophoresis, we've separated them. We transfer the protein to a nitrocellulose membrane. After we do Chihi, We transfer the proteins to a nitrocellulose membrane. Why am I transferring it? And then we block it with 5% BSA or milk. Just double check. Why are we transferring? Why transfer proteins to a nitrocellulose membrane? High nitrocellulose membranes are a popular matrix used in protein plotting because of their high protein binding affinity, compatibility with a variety of detection methods, and the ability to immobilize proteins and glycoproteins. Okay, I don't get that. One more time. Let's find something. Wow. Okay. So it sticks to the nitrocellulose because of the strong dipole. Okay. Strong dipole of the nitrate group in the MC membrane. And why do we want it to trans? Why do we want that? Following the separation, proteins are transferred to a membrane to immobilize the okay. So the separated proteins are then like separated, and you can do the antibody steps next. Got it. So you transfer them to a nitrocellulose membrane, and then you block with the 5% BSA or milk. Okay, I remember. So you block after. So you have the membrane now. Imagine a blanket. Those proteins are now there, and then you block it with the milk, so nothing will. So you get rid of the things you don't want, and then you incubate overnight with the primary antibodies, specifically for caveolin 1, flotilin 1, or GM1 ganglioside. And then you add a secondary antibody, hopefully that secondary antibody would have a fluorescent marker. So just for an hour, and then you wash off the secondary antibodies that did not bind to the primary antibodies. And then the enrichment, when you do chemoluminescence next, the enrichment of caveolin 1 and flotilin 1 and GM1 in those fractions, if you see them in those fractions using Western blotting, then you know that your lipid raft isolation was successful. Okay, good. So, what's next? Lipids. Cell membrane chemical composition. So lipids, you have a lot of different phospholipids, okay? So lipids, so the cell membrane has different. You have, no, one more time. The cell membrane chemical composition is a mouthful. You have lipids, proteins, and carbohydrates. And for lipids, you have phospholipids, glycolipids. No, phospholipids, glycosphingolipids, cholesterol, PGC, phospholipids, glycosphingolipids. For phospholipids, you have specific ones also. You have phosphatidylethanolamine, phosphatidylserine, phosphatidylinositol, phosphatidylcholine, sphingomyelin, and glycosyl phosphatidylinositol, and so on. Okay, so a lot of different stuff. So what lipid makes up 70% of the membrane? You see, not PASOCO, not personal computer. It is phosphatidylcholine. Phosphatidylcholine makes up 70% of the membrane because it is the most abundant phospholipid in eukaryotic cell membranes. Okay, I have to find a sticky note for this. 1 second. Found it. Alright, it's here. So what lipid makes up 70% of the membrane? Phosphatidylcholine. What does it look like? It is composed of choline, phosphate, glycerol, and fatty acids. So choline, yeah, I have to zoom in with my eyes. Choline, phosphate, glycerol, and fatty acids. For the polar head or hydrophilic head, it's the choline, phosphate, and glycerol. Meanwhile, the fatty acids are the non-polar or hydrophobic tails. Okay, got that one. So why is cholesterol important? Why? Why is it important? It's important because it modulates membrane fluidity and stability, ensuring proper function of membrane proteins and signaling pathways. And it makes up about the 20 to 25% of total lipids. Fluidity and stability would be the keywords for cholesterol. It's like it's a saling pusa. It's gonna be in the middle of the things. Okay. Next, biosynthesis of glycosphingolipids. Okay, from ceramide. So, from ceramide is glycosylated by glucosyl ceramide synthase with UDP glucose to form glucosyl ceramide or these glycolipids act as precursors for complex. Okay, that part is important. These glycosphingolipids act as precursors for complex glycolipids, playing essential roles in cell signaling, membrane stability, and lipid rafts. Gangliosides. They would differ in... So you have gangliosides. Okay, it's a class of glycosphingolipids with a ceramic core. Ceramide core. Then next, so the most common lipid in the cell membrane, phosphatidylcholine, and then cholesterol structure. Yeah, there it is. I found it. In the cell membranes, cholesterol inserts between the phospholipids with its polar head aligning with the phospholipid head groups, while the ring, while its rigid ring structure stiffens the upper membrane region. And so, yeah, it's for the fluidity. And in the cell membrane, what's the lipid to protein ratio? Okay, I feel like this one's important. RBC, myelin, and mitochondrion. RBC is 1:1. The lipids to proteins, it's a balance between flexibility and proteins for structural support. Meanwhile, myelin, I would imagine it's 4:1. It has more lipids. Why? Because it provides electrical insulation, which would reduce ion leakage for fast nerve signal transmission. And for mitochondrion, it's 1:3. You have more proteins because it's packed. It's packed with enzymes and transport proteins, and you need it for ATP production by oxidative phosphorylation. So RBC, it's 1:1 lipid protein. Myelin, a gazillion more lipids, 4:1. So because you need it for insulation to ensure the transmission of nerve signals. Meanwhile, for mitochondrion, you have 1:3. You have a lot more proteins for ATP production via oxidative phosphorylation. So, characteristics of the cell membrane. Oh, there is a you have a bit. Let's start with fluidity. It's very fluid. Well, it depends. Well, so with a cell membrane, you have cell membrane fluidity, which would highlight the lipid movement and some evidence for that. It exhibits lateral diffusion, flexion, and rotation. Again, what does it do? Lateral diffusion, flexion, and rotation. Flip-flop is rare. Lateral diffusion would be, I would be lying down on the ground and just ligid, like left and right, left and right. That's lateral diffusion, I think. No, never mind. No. That's rotation. 1 second. So lateral diffusion, I'm just looking. I imagine it's like a jellyfish. Like that. Just floating left and right. There you go. Flexion would be kind of like this. That kind of left and right. Meanwhile, rotation would be the ikot-ikot part. Okay. It's hard if you don't have a 3D model to demo it. But in my head, it should make sense. So lateral diffusion is you just float left and right. Flexion is like bending left and right. Fluid versus viscous or viscous. Okay, unsaturated fatty acids increase their fluidity by preventing the tight packing. You have the unsaturated hydrocarbon tails with the mini kinks, but the saturated fatty acids make the membrane more rigid. And then what is this? The human mouse cell fusion assay. It demonstrates the membrane protein mobility. How? So first you have the mouse and human proteins segregated, but after incubation, they would mix. Okay. At first, the human and mouse proteins are like segregated, but then you incubate them at 37 degrees Celsius, not Fahrenheit, Celsius. They would mix due to membrane fluidity. Okay. And then we have FRAP, fluorescence recovery after photo bleaching. What is that? I don't know. We should know. So fluorescence recovery after photo bleaching. It's used to study membrane fluidity and protein mobility. And in this method, membrane proteins or lipids are labeled, fluorescently labeled, and a specific region is bleached with a laser, causing the fluorescence loss. And over time, the unbleached fluorescent molecules would diffuse into the bleached area, and then you get fluorescence recovery. I see. I get it. Okay. So, if it's like Attack on Titan, why am I doing this? So, if it's Attack on Titan, and I imagine, no, not that one. Let's use the Shinra Tensei example in Naruto Shippuden. If Pain or Nagato does his Shinra Tensei, that's the bleach part. And then all of the villagers in Konoha, like a part of them would get bleached and now have white hair. But then they would just all like keep on walking around and diffusing and like going around. And soon enough, that very bleached or damaged part will be gone and will recover. So that is fluorescence recovery after photo bleaching. Next, asymmetry. So there is lipid bilayer asymmetry where, so this is where the PS and PE differ. Hold on. So you have a lot more. So imagine this. There's the extracellular space, and there's the cytosol, and there is the phosphatidylserine and phosphatidyl ethanolamine. They're outside. PS and PE are outside in the extracellular space. Meanwhile, PC and S. What? No, it's the opposite way. Where is my eraser? It's the extracellular base and cytosol. Why is that different? Outer leaflet. I have to make sure I get that part. Who is inside and who is outside? So, cytoplasm. Outer leaflet. Okay, so cytoplasmic. PS and PE are inside. No? Wait. Google. Where is... I don't want to get this confused. Where does phosphatidylserine not... Where is it found from? Inner cytoplasma membrane, inner leaflet of the membranes. So it's inside. But there's a difference between inside or very, very inside. So I just want to check. I think we can clarify that in a bit during the apoptosis part. Soon. Okay, apoptosis. Wait, here. There's a sentence. Annexin V binds to PS, detecting apoptosis when PS flips to the outer membrane. So it's supposed to be inside. It's supposed to be inside the membrane. PS and PE are in the cytoplasmic leaflet. But if PS starts to get exposed to the outer membrane, annexin V can bind to it, which would be an indicator of apoptosis or cell death. Okay. So what would regulate the different lipid distribution? That is flipases, flopases, and scramblases. Flipases, flip. You move the PS and PE inward. Flipases. Flopases, out. Where is my inside and outside? Okay, yeah. PS and PE inward. Scramblase will just mix them randomly. Flipases. And yes. Note, scramblases, they are calcium-dependent enzymes that randomly redistribute lipids. So calcium-dependent. Okay, there you go. Found the page. How do scramblases and flipases work together? This will clarify my question earlier. So there is a coordination between scramblases and flipases in regulating membrane asymmetry, especially during cell death, apoptosis, I said. And in normal, we're talking about normal conditions. In normal conditions, the flipases are active and they use ATP to maintain PS into the inner leaflet. Meanwhile, while the scramblases are like inactive. So we're just talking about the normal situation wherein the flipases would keep on bringing the phosphatidylserine into the inner leaflet via like it'll use ATP as to power it. But upon cell death activation, the scramblases would be active and the PS can just be thrown outside while the flipases would be inactivated, and the PS exposure is a key apoptotic signal for immune clearance. Okay, so let's talk about that again. Normally, the flipases would have, or normally the flipases would be carrying the phosphatidylserines inside the leaflet using ATP as a power up or a quirk. But then upon cell death activation, the scramblases would be active while the flipases are inactive. Scramblases would just throw them outside, and the PS or phosphatidylserine exposure is a key indicator of apoptosis. Okay. And how does phosphatidylserine exposure contribute to apoptotic cell clearance? Well, once they are exposed or outside, that is an eat me signal, allowing the macrophages and other immune cells to recognize and engulf the dying cell. So once the phosphatidylserine is outside, that's technically the flag. They're like, kill me now, like that. And the macrophages will just eat them up. Next. So how the plasma membrane asymmetry affects function? The left side, well, I can see the picture. I'll just describe it. So the left side would be resting state, where the membrane lipids and proteins are asymmetric. But when the antigen binds, structural changes would occur, and this lipid asymmetry shift would lead to receptor clustering and etc. There's also the bile canaliculus or Philippine staining. Bile canaliculus asymmetry in hepatocytes using Philippine staining, and that would detect cholesterol-rich membranes. So normally, the bile canaliculus is a special apical domain for bile secretion and cholesterol transport, and it would demonstrate membrane asymmetry between apical and basal surfaces. And this polarity ensures that bile components are actively... Okay, I got lost. One more second. I know that it's crucial for bile secretion and transport. Membrane asymmetry between apical and basal. Okay. And this polarity would ensure that that bile is really transported into the canaliculus, while the nutrients are pushed to the basolateral side. Okay. So the Philippine staining is a fluorescent dye-based method used to detect cholesterol in biological membranes. Philippine staining would detect cholesterol. Philippine staining would detect cholesterol. Philippine staining would detect cholesterol. It's an antibiotic that binds specifically to unesterified or free cholesterol. And the fluorescent complexes can be visualized under a fluorescence microscope. Okay, so it would bind to the esterified or free cholesterol and can be visualized using a fluorescence microscope. Key uses of Philippine staining: you have cholesterol localization, which identifies cholesterol rich domains. That's the first. And then you have finished. 6 ways the membrane proteins associate with the lipid bilayer. Good one. So you have transmembrane or single pass, transmembrane multipass. So the single pass, you have a single alpha helix spanning the bilayer. It's one like that. Meanwhile, the multipass would be like you go up and down, The multiple alpha helixes would traverse the membrane, which could form channels or transporters. You also would have lipid anchored cytosolic side attached via the lipid tails, and embeds the protein in the inner leaflet. You also have the exoplasmic side, cytosolic. Okay, that's a lot of different stuff. Peripheral membrane protein associate with the membrane without embedding into the bilayer. They could attach via phospholipid charge groups or other proteins. How? So you have the protein kinase C, which binds negatively charged phospholipids in a calcium-dependent manner. How? Phospholipid charge groups. Protein kinase C, which binds negatively charged okay. And peripheral proteins can bind into the proteins. Okay, so just using other proteins. So charge group of proteins. Overview of lipid-linked proteins. You have the G proteins associated with some receptors. This one's very specific. I might talk about this also later. Where am I now? Still in the nice. So you have G proteins. It's a family of guanine nucleotide binding proteins which act as molecular switches inside the cell. And they play a crucial role in signal transduction, yes, relaying the signals from GPCR or G protein coupled receptors to intracellular pathways. And the slide would mention the adrenaline and noradrenaline receptors, but those are just examples. Structures of G proteins. You have G alpha subunit, which has the GTPase domain. And what is that? That will hydrolyze GTP and GDP, which will determine if it's active or inactive. Meanwhile, the G beta and G gamma subunits will just form a stable complex. And some G proteins are lipid linked, meaning they have lipid modifications which will help them attach to the membrane. G alpha unit is linked to the membrane via the geranyl group at the N terminal. Okay. G gamma subunit is linked via the myristoyl group at the C. I have to write that. Those are very different words. G alpha, the N terminal, that's the geranyl group. Meanwhile, G gamma, it's linked via the myristoyl group. Meanwhile, the G beta subunit, it's not lipid linked, making it very effective as a peripheral protein. And so when GPCR is activated by a ligand, such as adrenaline, it would induce a conformational change, which activates the G protein via the exchanging GDP to GTP on the G alpha subunit. G alpha subunit would then dissociate from the G beta gamma complex, and that would allow each to interact with different effectors. And then that is an example with that would be, what is an example of that? And such as, okay, some specific risk effectors are adenylyl cyclase, phospholipase C, or other ion channels. And this would just cascade the signal. Membrane carbohydrates, you have proteoglycans, which is a subset of glycoproteins where the carbohydrate component is glycosaminoglycans. What is the cell coat? The cell coat or glycocalyx, it appears as a fuzzy dark layer on the outer cell membrane if you use an electron micrograph. What? If you use an electron micrograph. Okay, next is, where is that? Labels would show the plasma membrane and cytoplasm. Let me just check something. Ah, okay, I got it. Let me just adjust something real quick. This is, I see. There you go. I got it. So that was that sound. Okay, moving on. So the electron micrograph, it would appear as a fuzzy dark staining layer on the outer cytoplasm or outer plasma membrane. Illustrates the glycocalyx's molecular component. So that's the cell coat. So what's the cell coat? It is a thick carbohydrate layer of glycoproteins, proteoglycans, and glycolipids. And there's different sugar residues. Next, red blood cell membrane. So that's a very specific membrane also. How do we know membrane asymmetry exists? You could do a couple of things. Ooh, different experiments again. Maybe I should have a very specific page for this one. Like for just the different pages. I mean experiments. Yeah, I should. Let me find a piece of papers. Found one. Okay. So what I was saying a couple of things earlier. I just need to write those down. Which experiments did I say? We have lipid raft isolation, membrane experiments. You have the lipid raft isolation. Raft isolation. Oh, this one's funny. Second, no. The lipid raft isolation, that would just end with the Western blot. But I will also blot. Sticky note will be green for experiments. Green. Next. I think there was another one. It was the bleach. What page? Okay, there you go. Found it. Almost lost it, but it is found. Fluorescence recovery after photo bleaching. Okay, got it. And then we had, what did we have? Philippine staining. Oh, Philippine staining. I think I have to turn on aircon. It's on. What else? Next would be the RBC ghost experiments, but we did not talk about that yet. So let's do it. Okay. It's the RBC ghost experiments. You would use protein labeling and glycolipid sugar hydrolysis to study the membrane composition. And there is also freeze fracture electron microscopy. So how do you prepare the RBC ghosts? You first do hypotonic lysis, which would remove the hemoglobin, and that would leave only the membrane. So hypotonic lysis removes the hemoglobin, leaving only the membrane. And various treatments would produce leaky ghosts, sealed ghosts, right side out vesicles, and inside out vesicles. Okay. And then the markers. This is the important one for lipid analysis. Annexin V would bind to the PS, the phosphatidylserine. Crucial because flipases normally bring them inside, but during apoptosis, the scramblelases would put them out. And then now that they're outside, that's a signal of your cell. They're like, I'm gone, I don't want to be here, exclude me from Earth. And so the macrophages will just take them in. Okay, I have to write down for outer leaflet and inner leaflet. What's the difference? We see 70% of lipids in the cell membrane. Cell membrane. Got it. Next, types of membrane transport. This one might be more familiar for a lot of people. So that's the simple diffusion, which could be. Wait, so let's start with passive and active transport, okay? So passive transport, no energy required. Active transport, energy is required. And for passive transport, you could do simple diffusion where the nonpolar molecules could just move directly across the membrane, small nonpolar. And you would also have channel mediated transport, aka ion channels, allow the specific ions to pass through. Transporter mediated transport, you have the carrier proteins facilitating the movement of larger molecules such as glucose. Meanwhile, the active transport would need to use ATP because you're pumping molecules against their gradient. So, for example, sodium transport would require energy input. What are the types of transporters? You have the glucose, which is the uniport, symport. Wait, 1 second. Types of transporters, you have 3: uniport, symport, antiport. Uniport moves a single molecule in one direction. Symport is 2 molecules in the same direction. Antiport, 2 molecules in different directions. So those are coupled transports, the symport and antiport, but just different directions. So again, types of membrane transport. You have passive and active. Passive, you can just have normal diffusion. Channel mediated and transporter mediated. Active would need ATP. Types of transporters, you have uniport, symport, antiport. Uniport, one way, only one. Symport is 2, same direction. Antiport is 2, different directions. What are some different factors restricting membrane protein mobility? Well. Some proteins are anchored to cytoskeletal elements such as inside the cell or the extracellular matrix outside the cell. So that will really really hinder their mobility. And some proteins aggregate into specific regions, forming distinct membrane patches, such as lipid rafts and coated pits and gap junctions. So lipid rafts, that's rich in cholesterol and sphingolipids, which is involved in signaling. Mentioned earlier, coated pits would be sites for receptor-mediated endocytosis, and gap junctions would be for cell-to-cell communication. Okay. Membrane skeleton and protein anchoring. What is the membrane skeleton? Is a cytoplasmic filament network that maintains the cell shape and restricts protein movement. And some key structural components would be spectrin, ankyrin, actin filaments, and membrane proteins. Spectrin forms a scaffold beneath the membrane. Ankyrin links the spectrin to transmembrane proteins. And actin filaments support the cytoskeletal connections. Spectrin, ankyrin. Spectrin is below. Ankyrin links the spectrin to the transmembrane proteins. I cannot flip this page. Okay, there. Impact of membrane lipids on function. Oh, a lot of impact. So you have adipocyte plasma membrane, GM3 levels, and insulin resistance. So normal adipocytes, normal adipocytes. You have insulin signaling occurring in the lipid rafts. GM3 interacts with the insulin receptors. And then you have the signaling via the IRS, PI3K, and GLUT4. And then you have the glucose uptake. But if you have insulin-resistant adipocytes, the inflammatory signals, such as TNF-alpha, would increase. The GM3 would relocate from the caveolae, the mini bowl things earlier, from the caveolae to the mobile GM, and this would disrupt the IR function. What IR function? Maybe insulin resistance? Okay. Impairing the GLUT4 and contributing to insulin. Okay. So those are the different techniques. So a question could be, discuss the fluid mosaic model for biological membrane structure in terms of the component molecules and explain how modifications in composition can affect the ability of molecules to move within the membrane and across it. So you have the fluid mosaic model, and it essentially describes how the plasma membrane has different proteins and lipids inside and carbohydrates as well, and it's fluid because it will just flow. It is the lateral movement of the different components, and some components will be the phospholipids, proteins, carbs, and the effect of the composition. For example, if you have a lot of cholesterol, it would decrease the permeability and increase the fluidity. If you have more saturated fatty acids, it would be very, very rigid. If you have more unsaturated fatty acids, it would have more kinks and would increase the fluidity. And if you have a lot of lipid rafts, that would restrict the protein mobility, and because you have the microdomains. Next, compare and contrast different forms of proteins associated with membranes. Some different forms would be transmembrane proteins and lipid anchored proteins. You have the peripheral membrane proteins. So the transmembrane proteins would span the entire layer. Most of its function would be transport, signaling, and enzymatic activity. You have the lipid-anchored proteins, which are covalently attached to lipids in the bilayer. You also have the non-embedded peripheral membrane proteins, just interact with the integral proteins or lipid head groups via electrostatic forces. And then you have an example of the peripheral membrane protein would be spectrin. Spectrin is a part of the cytoskeleton which maintains the red blood cell shape. Spectrin is a part of the cytoskeleton which maintains the red blood cell shape. Some interactions for the lipid bilayer would be hydrophobic interactions stabilized by hydrophobic amino acids interacting with lipid tails, and electrostatic, which would mean interacting with charged heads. Permeability, so account for biological properties of the membranes. Okay, permeability, movement of molecules across the membranes with reference to the structural properties of the proteins and their location in the bilayer. So for permeability, the small nonpolar can just diffuse quickly or freely. The small uncharged polar will be slow. They will diffuse slowly. The large uncharged polar would need the transport proteins, while the ions would need the bumps or channels. Passive transport, active vesicular would be bulk. And nature of specialized domains, you have the lipid rafts. Okay, I mentioned that one earlier. Flow cytometry. Ooh. Staining with confocal. So if I was design an experiment to determine whether a receptor is located in the lipid raft or non-raft region. So we have the estrogen receptor. We want to test it. I would check first using like Western blot after the cold detergent extraction mentioned earlier, because they are detergent resistant and will not solubilize. So use the Triton X, lyse the cells with Triton X, centrifuge, load the supernatant onto the gradient, and centrifuge it even more. And then the raft is the super low density at the top. And then I would then Western blot to check for the raft markers. And if the ER marker is in the low density region, then it's in the raft. But if it's not, then it's in the non-raft. Flow cytometry would just do quantification of it. So flow cytometry with cholera toxin B and then ER antibody. I'm not sure about this one. That's fine. There's also immunostaining with confocal. So fix and permeabilize, stain for the lipid rafts using the anti-caveolin 1 antibody, because caveolin 1 is in the lipid rafts. If you stain for it and use confocal, hopefully you'll see it. And then qPCR would measure the mRNA levels in raft versus non-rafts. So if the mRNA decreases in raft and increases in non-raft after, that's a bit confusing. It's fine. Next is cell junction and polarity. Cell junction and polarity. Should I save it? Okay. I think that's all for today. Thanks for watching. Bye.